You know, there is a specific kind of existential dread I get when I look at the material we're covering today.
I
It's a
did
heavy stack.
dread. That is not the reaction I usually get for neuroscience deep dive. Usually it's confusion or maybe intimidation, but dread.
It is dread.
Hmm.
Because we are looking at chapter 1 of what is arguably the Bible of neuroscience. And the central thesis, the thing we're going to spend the next hour proving,
Mm.
is that everything I am, everything you are,
Hmm.
the fact that I love my kids, the fact that I can appreciate a sunset, or even the fact that I'm, anxious about recording this, all of that is just meat. It's just wet biology.
It is a strictly materialistic view, yes. It strips away the magic, or at least it attempts to strip away the supernatural element of the self.
Exactly. It's the idea that the mind, this floaty, ethereal thing we think we have, is actually just a set of operations carried out by the physical brain. There is no ghost in the machine.
There's just the machine.
And for some reason, that freaks me out.
Well, it's a fascinating subject because it really challenges the way we all view ourselves. But you have to remember, getting to that conclusion that the mind is the brain was a wild ride. It wasn't a gentle realization.
Right.
It was a centuries-long argument involving some of the most stubborn and brilliant people in the history of science.
So that is our mission today. We
Yeah.
are going to track that war. We are going to move from the cellular level, the nuts and bolts, all the way up to the anatomy, the history of how we mapped this thing, and finally, to the complex stuff,
Right, like language, emotion, and the big one.
consciousness.
Precisely. The goal is to answer a really fundamental question. How do billions of individual nerve cells produce behavior? How do we get from biology to psychology?
And to understand that, we have to look at the tension that runs through this entire story,
The great battle.
the battle between localization and holism.
Right. Does a specific part of the brain do a specific thing? Or does the whole brain work together to do everything? That question is the engine of the whole story.
So let's start at the bottom, the building blocks. The source material takes us back to a time when we didn't even really know what the brain was made of. I want to start with the fluid era because this just blows my mind.
You mean Galen?
I mean Galen,
Yeah.
2nd century Greek physician.
Mm-hmm.
This guy's shadow is so long, it basically covered medicine for, what, a millennium and a half?
At least. His ideas were dogma for 1,500 years.
And he didn't think the brain was electrical. He thought it was hydraulic.
Well, you have to put yourself in his shoes, right?
Huh.
He's dissecting brains, mostly animal brains, sheep, some apes, and what does he see? He cuts into the brain tissue and he finds these large, empty spaces inside.
The ventricles.
The ventricles, exactly. And in a living brain, those are filled with cerebrospinal fluid. So he sees this.
So he looks at this wet sponge and thinks, okay, the solid part isn't doing the work. The liquid is.
Right. It's a pretty logical leap for the 2nd century. He believed that the nerves were hollow tubes, like pipes. And the brain's job was to pump this psychic pneuma, this vital spirit fluid, down the tubes to inflate the muscles.
So when I flexed my bicep, Galen thought I was literally pumping it up with brain juice.
Essentially, yes. Your body is a hydraulic puppet show, and the brain is the pump operator. And because they didn't have microscopes, nobody could prove him wrong.
Right, because if you cut a nerve, it doesn't look hollow to the naked eye.
Not at all. But the argument was always, well, maybe the tube is just too small to see. That idea, that sort of hydraulic theory, it stuck around all the way until the industrial age. It's an incredible example of intellectual inertia.
It's amazing how long a wrong idea can survive, if it's intuitive. But the wall finally comes down in the late 1800s. We finally get the microscope technology to see the actual texture of the brain.
Yes.
And this sets the stage for the heavyweight championship of neuroscience. Golgi versus Cajal.
This is the defining rivalry of the field, without a doubt. Camillo Golgi, the Italian, and Santiago Ramon y Cajal, the Spaniard.
And they hated each other.
They were not friends, which
Yeah.
is just so ironic because they ended up sharing the Nobel Prize in 1906 for this very work.
That must have been an awkward dinner.
I can't even imagine. But the tension came down to a fundamental disagreement about what the brain actually is at a microscopic level.
Before we get to the fight, we have to talk about the weapon,
The
because Golgi invented it, something called the black reaction.
Reazione Nera. This was in 1873. And you have to understand, before this, if you looked at brain tissue under a microscope, it was useless. It just looked like a tangled mess.
Like a forest where all the trees are growing into each other.
That's a great way to put it. You couldn't tell where one cell ended and the next began. The brain is just packed. So incredibly tight.
Too dense.
Way too dense. But Golgi discovered, almost by accident, that if you treat the tissue with potassium dichromate and then silver nitrate, something very strange happens. The silver crystallizes inside the cells, but, and this is the crucial part, it doesn't stain all the cells.
It only stains a tiny fraction, right?
About 1 to 3%. And it's completely random. So instead of seeing a solid black blob, you get these beautiful isolated silhouettes of individual neurons against a clear yellowish background.
Wow.
For the first time, you could see the entire structure. The cell body, the long axon coming out of it, the complex branching dendrites, everything.
It's like turning off the lights in a crowded room and hitting just 3 people with a spotlight.
Yeah.
Suddenly you can see their shape, their posture, everything without the crowd getting in the way.
That is the perfect analogy. So Golgi invents this revolutionary tool. He looks at the brain, and he makes a massive fundamental mistake.
He sees the syncytium.
That is the term, a syncytium. He looks at all these tendrils, the axons and dendrites, and he decides that they are all physically fused together. He thinks the brain is like a giant fishing net, a continuous web of protoplasm.
So he didn't believe in individual cells.
Not in the nervous system. He thought it was one giant connected meshwork. This is called the reticular theory.
Which feels almost like he's trying to hold on to that old hydraulic idea in a way.
Yeah.
If it's all connected, fluid or signals can just flow anywhere.
It was a very compelling idea to the scientists of the time. It implies a kind of holistic unity. But then you have Kajal. And Kajal is, well, he's a shark. He's relentless. He takes Golgi's own invention, this silver stain, and he uses it to completely destroy Golgi's theory.
I love that. Thanks for the tool. I'm going to use it to ruin your career.
Kajal was just a better observer. And he was a brilliant artist. His drawings of neurons are still considered masterpieces. He improved the staining technique, made it more reliable, and he did something very clever.
What's that?
He looked at younger brains, embryonic birds and mammals where the meshwork wasn't as dense yet, the forest wasn't so overgrown, and he saw something that Golgi had missed.
The gaps.
The gaps. He saw that the axon of one cell would reach out and get incredibly close to the dendrite of another, but they didn't fuse. There was a tiny physical separation, a space.
So the brain isn't a net.
It's not a net. It's a network of billions of discrete autonomous units, individual cells.
And this is the birth of the neuron doctrine.
It is. The principle that the individual neuron is the elementary building block and the fundamental signaling element of the entire nervous system.
But remember, back in the 1890s, Kajal couldn't actually prove the gap existed, could he? His microscope wasn't strong enough to see something that small.
He couldn't. It was an inference. He just intuited it from the shapes he was seeing and drawing. So for a long time, it was just his word against Golgi's. It was a huge debate.
And Golgi was the establishment figure.
Oh, very much so. And he never, ever backed down. Even at his Nobel Prize acceptance speech, with Kajal sitting right there in the audience, Golgi went up there and defended the reticular theory. He basically told the assembled scientific community, this other guy sharing the prize with me is wrong.
That is incredibly awkward. I can't even imagine the tension in that room. So when did we actually know for sure who was right?
It took a few more decades. We really needed 2 more pieces of evidence to put the nail in the coffin of the reticular theory. The first piece came in the 1920s with an American embryologist named Ross Harrison.
The tissue culture guy.
That's him. He did something absolutely brilliant. He took live nerve cells out of a frog embryo and put them in a tissue culture. Basically a little petri dish with all the nutrients a cell needs to survive.
So there's no brain, no body, just the cell floating in a dish.
Just the cell. And he watched it with a microscope for weeks. And he saw it sprout. He literally watched the axon grow out of the cell body, extending this little hand-like structure at its tip.
The growth cone.
The growth cone, yes. And it was actively searching, feeling its way forward, looking for a target.
Which proves it can't be a pre-existing web. If I can watch a single cell grow its own arm, even when it's totally alone in a dish, it has to be an independent entity.
Precisely. It was the smoking gun for the autonomy of the neuron. The cell is the basic unit. But the final proof, the actual photograph of the gap itself, that didn't come until the 1950s.
The electron microscope.
Right. Sanford Pillay, using one of the first electron microscopes, which uses a beam of electrons instead of light and has wildly higher resolution, finally took a picture of the synapse.
And there it was, the empty space.
The synaptic cleft. It's tiny, about 20 nanometers wide, but it's there. The neurons don't touch. The web theory was officially dead. Kajal was right.
Okay, so we've established the hardware. We aren't a bag of water, and we aren't a fishing net. We are a galaxy of individual stars, these neurons, separated by empty space. But, I mean, that creates a whole new problem.
It does.
If they don't touch, how does the message get across?
That is the perfect pivot to the second half of this puzzle. If it's not water pressure and it's not a direct physical connection, what is it?
It's electricity. But not just electricity.
Exactly. We had the frog leg guy, Luigi Galvani, way back in the 1700s.
Right, the lightning experiment.
He showed that if you apply an electrical current to a dead frog's nerve, the leg kicks. So we knew nerves had an electrical component. They were like wires. But if they were just simple copper wires, the signal would jump that synaptic gap at the speed of light.
And Helmholtz, who I think of as the speed limit cop of neuroscience, proved it wasn't that fast.
Hermann von Helmholtz, a titan of 19th century science, he actually set up an experiment to measure the speed of the nerve impulse. And he found it was, well, slow.
How slow?
About 30 to 90 meters per second, which sounds fast, and it is. It's like a fast race car, but it's nowhere near the speed of light. It's millions of times slower.
So something else is happening. There's a delay. It's not just a spark jumping a gap.
Exactly. There's a process.
Yeah.
And that's where the chemistry comes in. This is the realm of pharmacology. People like Claude Bernard, Paul Ehrlich, and John Langley in the late 19th and early 20th centuries.
What were they doing?
They started realizing that if you put certain drugs like Curair, the South American arrow poison, on the junction between a nerve and a muscle, the muscle stops responding to the nerve.
But the muscle itself still works if you shock it directly, right?
Yeah, exactly. The muscle is fine, the nerve is fine, but the message isn't getting through at that specific junction point. This suggested that there had to be a specific receiver, what they started calling a receptor, at that junction that the poison was blocking.
Ah, so the poison molecule gets there first and sits in the parking spot, so the real message can't park.
That's a great analogy. So the model becomes an electrical signal travels down the wire, the axon. When it reaches the end, it causes the release of a little packet of chemicals neurotransmitter.
The chemical packet swims across the gap.
It swims across the synaptic cleft, finds a specific receptor on the next neuron, and binds to it.
And that binding starts a new electrical signal in the second cell.
You've got it. It's an electrochemical transducer. And that complexity, that two-step process, is what allows the brain to be so much more than just a simple reflex machine. It allows for modulation, for inhibition, for learning, for thought.
So we have the building blocks, we have the cells, we have the signals. Now we have to zoom out, because the biggest fight in the history of neuroscience wasn't about cells. It was about the map.
The great debate. Localization versus holism.
And this brings us to a guy with a very complicated reputation, Franz Joseph Gall.
Gall is a fascinating figure. He was working around 1800, and you have to give him credit. He proposed 3 radical ideas that were, in principle, absolutely correct.
Okay, let's hear the hits before we get to the misses.
First, he argued very forcefully that the brain is the organ of the mind. That wasn't a given back then. People were still debating if it was the heart or the liver.
Okay, that's a big one.
Huge. Second, he said the brain is not a single organ, but a collection of at least 27 specific organs or faculties. And third, and most importantly, he said that these specific regions control specific functions.
That sounds remarkably modern. That's the foundation of modern neuroscience.
It is prescient. But his method for finding those regions was, well, it was phrenology.
Skull bumps.
The skull bumps. Gall's fatal flaw was that he didn't look inside the brain. He only looked at the surface of the skull. His theory was that if you exercise a mental faculty-like memory, or amativeness...
Amativeness, what's that?
Romantic love, basically, the capacity for it. He argued that if you use that faculty a lot, the corresponding brain region grows, like a muscle. And because the skull is hard, that growing brain pushes the skull out, creating a bump you can feel.
So if I have a bump behind my ear, that means I'm good at math or something.
That was the logic, yes. There's a funny anecdote in the source material. Gall said he formulated this whole theory because he noticed that his classmates in school who had really good memories all had prominent, bulging eyes or eyebrows.
Okay.
So he concluded that the memory center must be right behind the eyes, in the frontal lobe, and that in people with good memories, it had grown so large it was literally pushing their eyes forward.
That is distinctively not the scientific method. That's just seeing a pattern and making up a story.
It is not, and that lack of rigor is what ultimately sank him and turned phrenology into a pseudoscience, a parlor game. But the core concept of localization was out there. However, the scientific establishment pushed back hard.
Enter Pierre Florent.
The rebuttal. Florence was a respected French physiologist in the 1820s. He decided to test Gall's ideas experimentally, not just by looking at skulls.
And his experiments involved
Pigeons.
pigeons and rabbits, yes. He started systematically destroying or ablating regions of the brains in these animals.
That sounds pleasant.
It's the history of science. It's rarely pretty. But his results were interesting and, at the time, seemed conclusive. He found that it didn't seem to matter where he made the lesion in the cerebral hemispheres.
What do you mean?
Removing a piece from the front, the back, the side, it all seemed to have the same general dampening effect. It weakened all the cognitive functions equally. The more he removed, the weaker the animal got, but no specific function disappeared.
So he concluded.
He concluded that the brain functions as a whole. This is the aggregate field view, or holism. He famously wrote, "All perceptions, all volitions occupy the same seat in these organs. The mind is a single, unified property of the whole brain."
And I imagine this was popular with the religious crowd. Extremely popular. The cultural context here is super important. Gall's view was seen as dangerously materialistic. It broke the mind into biological chunks. Floren's view seemed to preserve the idea of a unitary soul.
Right, if the brain is one big, undifferentiated mass, it's easier to view it as the vessel for a single, indivisible spirit.
Exactly. It was less threatening to the established order. So for a while, holism wins round one. Localization is out, aggregate field theory is in, but then comes the turning point, and it comes from studying language.
This is the moment everything changes.
Yeah.
1861, Pierre-Paul Broca.
A French neurologist. And his famous patient, a man named Louis-Victor Le Bon,
But they called him Tan.
they called him Tan because that was the only syllable he could say, for years.
Tan.
Tan. He could understand language perfectly. If you asked him the time, he would point to the clock and know. If you asked him to perform an action, he did it. He had no paralysis of the mouth or tongue. He could whistle a tune. He could even sing a melody.
But he couldn't speak. He couldn't form words.
He could not produce grammatical speech. When Le Borgne died, Broca performed an autopsy and examined his brain. And he found a very specific localized lesion
Where was it?
in the posterior part of the frontal lobe, but only in the left hemisphere.
And Broca realizes.
He realizes that this specific spot must be the control center for the production of speech. He went on to study 8 similar patients, all with the same issue, all with lesions in the exact same spot on the left side.
That's conclusive.
It was. And he made the famous declaration at a scientific meeting. Nous parlons avec l'hémisphère gauche.
We speak with the left hemisphere.
This was the first true irrefutable evidence of localized function for a complex cognitive behavior. It wasn't phrenology anymore, it was clinical anatomy. The pendulum begins to swing back.
So Broca finds the speaking spot. But language is obviously more than just speaking, it's also understanding.
Right, and that's where Carl Wernicke comes in, about a decade later in 1876. He was a German neurologist, and he described a completely different type of aphasia.
Aphasia is just meaning a language disorder, right?
Yes, an inability to produce or understand language due to brain damage. Wernicke's patients were the opposite of Tan. They could speak. In fact, they spoke very fluently. Words would just pour out of them.
Fuck.
But the words made no sense. It was fluent gibberish, word salad. And more importantly, they couldn't understand what was being said to them. They were deaf to the meaning of words.
So Broca's aphasia is...
Yeah.
I understand, but I can't speak. Wernicke's aphasia is... I can speak, but I don't understand.
Exactly. And when Wernicke looked at their brains, the lesion wasn't in the frontal lobe like Broca's patients. It was further back, in the posterior part of the temporal lobe, where it meets the parietal and occipital lobes.
So now we have 2 distinct spots, one for talking, one for comprehending.
But Wernicke went a step further, and this is what makes him a genius. He didn't just say, here are 2 spots. He proposed a network. He formulated the first coherent model of distributed processing.
Unpack that. What does that mean?
He argued that complex behaviors like language aren't just in one blob of tissue. They arise from the interaction between these different elementary processing units. He actually outlined the circuit.
Like a wiring diagram.
Exactly, he said. Input comes in, let's say, auditory input. It goes to Wernicke's area first to get meaning to be comprehended. Then that information travels along a pathway to Broca's area, which puts it into a grammatical structure and creates a motor program for the mouth and tongue. And then that program is sent to the motor cortex, and you speak.
So it's a factory line. Comprehension, then grammar, than output.
It is. And Wernicke made an astonishing prediction based on this model. He said, if there is a wire connecting these two areas, which we now know is a bundle of fibers called the arcuate fasciculus, and if you cut that wire, you will get a third distinct type of disorder.
He predicted a disease before he'd ever seen a patient with it.
Yes. He called it conduction aphasia. He said the processing centers themselves are fine, you can understand speech perfectly, and you can physically speak perfectly, but the bridge between them is out.
So you can't.
You cannot repeat words. If I say apple, you hear it and you understand what an apple is. But you can't send that information from the comprehension center to the speech production center to say the word apple. The message gets lost in transit.
That is incredible. And of course, they eventually found patients with exactly that profile.
They did. And it really established the modern view, which is a synthesis of the two old ideas. It's not just localization, and it's not just holism, it's connectionism.
Mental processes are the end product of interactions between elementary processing units located in different regions.
That's the key takeaway. It's both localized and distributed.
Okay, before we move to the modern era and how we validate all this, we should probably do a quick tour of the machine itself. The source material gives us a grand tour of the central nervous system. I want to visualize this. If I'm a tiny person walking up the spinal cord into the skull, what am I seeing?
Let's do the tour. So the central nervous system, the CNS, has 7 main parts. Think of it as a hierarchy, moving from ancient basic survival functions at the bottom up to the most complex, evolutionarily new functions at the top.
Starting at the very bottom.
You have the spinal cord, that's the superhighway. It's receiving all the sensory information from your skin, muscles, and joints, and it's sending out the motor commands that control the movements of your limbs and trunk. It also handles all your basic reflexes.
So if I touch a hot stove, my spinal cord pulls my hand back before my brain even knows what happened.
Exactly. It's a local circuit. Now, as you move up from the spinal cord into the skull, you hit the brainstem. This has 3 parts. First up is the medulla oblongata.
Medulla is the life support system, right?
Absolutely.
Yeah.
Digestion, breathing, heart rate, blood pressure, all the stuff you don't want to have to consciously think about. If this gets damaged, it's game over.
Right above that.
You hit the pons. The word pons means bridge. It acts as a major relay station, specifically conveying information about movement from the cerebral hemispheres down to the cerebellum.
And the cerebellum is that wrinkly, cauliflower-looking thing at the back of the brain?
That's the one, the little brain. It's incredibly important. It doesn't initiate movement, but it modulates the force and range of movement. It's crucial for motor skills and procedural memory.
So if you are learning to play the piano or ride a bike, your cerebellum is working overtime.
It's what makes your movements smooth, coordinated, and accurate instead of jerky and clumsy.
Okay, we're climbing higher, above the brainstem.
You're in the midbrain. This controls many sensory and motor functions, but it's particularly important for eye movement and visual and auditory reflexes. Like when a loud noise makes you turn your head, that's your midbrain.
And above that.
We reached the diencephalon, which is Greek for between brain. This contains 2 huge critical structures. First is the thalamus.
The grand central station of the senses.
Perfect description. Almost everything you see, hear, or feel goes through the thalamus before it reaches the thinking parts of the brain, the cortex. It's the brain's receptionist, routing all the incoming calls. The only sense that bypasses it is smell.
And its partner.
The hypothalamus, which sits just below it. This tiny structure regulates homeostasis. It keeps your body in balance. It controls hunger, thirst, body temperature, and your circadian rhythms. It's the thermostat.
And finally, we get to the big boss, the cerebrum.
The cerebral hemispheres. The giant wrinkled outer layer is the cortex. All those wrinkles, by the way, the gyri and sulci, are just a way to cram a huge surface area into a small skull. If you flattened it out, it would be the size of a large pizza.
And this is divided into the 4 famous lobes. Let's run through them quickly.
Okay. Up front, you have the frontal lobe. This is the executive. Planning, decision-making, movement control, short-term memory. It's who you are.
Behind that.
The parietal lobe. This is for somatic sensation touch, temperature, pain, and for creating your body image and understanding where you are in space.
And all the way at the back.
The occipital lobe. This is one-trick pony. It does vision, and that's it. It's entirely dedicated to processing what you see.
And on the sides, by the ears.
The temporal lobe. This does hearing, but also higher order visual processing, learning, memory, and emotion.
And a key feature that we have to mention here is the contralateral control, right? The crossover.
Absolutely crucial. The left Cerebral hemisphere controls the right side of the body and receives sensation from it, and vice versa. It's a crossover system that happens down in the brainstem.
One name that pops up here when we talk about mapping is Brodmann.
Corbinian Brodmann.
Yep.
In the early 1900s, he was a German anatomist who did something incredibly tedious but important. He painstakingly looked at slices of the entire cortex under a microscope.
And he realized the cells weren't the same everywhere.
Exactly. He saw that the cortex isn't uniform. The arrangement, density, and type of cells change as you move from one area to another. It was like looking at a map of a city and seeing different neighborhoods with different types of houses.
And he drew a map based on that.
He mapped out 52 distinct areas based on this cytoarchitectonics, this cellular architecture. And we still use this map today. If I say Brodmann Area 17 to a neuroscientist, they know I mean the primary visual cortex. It was the first rigorous anatomical map of the cerebral territory.
So, okay, we have the neuron doctrine, we have Broca and Wernicke proving localization for language, we have Brodmann drawing the map. Case closed, right? Localization wins.
You would think so.
Okay.
But science is never a straight line. In the early 20th century, the pendulum swung back hard toward holism, and it was largely due to the work of one man, an American psychologist named Carl Lashley.
What was Lashley's beef with localization?
He was deeply skeptical. He thought these Brodmann maps and the case studies from Broca were interesting, but maybe not the whole story. He wanted more rigorous experimental proof, so he did an experiment.
The rat mazes.
The rat mazes. He would teach rats to run through a maze to get a food reward. Then he would make a lesion in the rat's cortex and see how it affected their ability to remember the maze.
And what did he find?
He found something very confusing. The severity of the learning and memory defect seemed to depend on the size of the lesion, not the location.
So it didn't matter where he cut.
That's what he claimed. He could cut out a piece of the visual cortex or the motor cortex or the sensory cortex, and as long as he didn't take out too much tissue, the rat could still eventually relearn the maze.
So he concluded.
He formulated a principle he called mass action, the idea that the whole brain mass works together in complex learning. He basically said, look, I cut out the visual area and the rat still learned the maze. Therefore, memory for the maze isn't localized in the visual area. He argued for a return to the aggregate field view.
God, was he right? Because that seems to contradict everything we just said about Broca and Wernicke.
He was profoundly wrong, and the flaw in his experiment is actually a great lesson in how the brain works.
What was the flaw?
Maze running is a multisensory task. It's incredibly complex.
So there are multiple ways to solve it.
Exactly. If you blind a rat by cutting out its visual cortex, it can still run the maze using its whiskers to feel the walls, or its sense of smell to follow cues, or its motor memory of the turns. The function of maze running wasn't unitary. The brain compensated.
So the brain has backup systems.
It has multiple parallel redundant systems. Lashley interpreted this compensation as the whole brain works together, but really it was the brain has multiple different tools it can use to solve the same problem.
It's like if I tell you to get to the grocery store and I block the main highway, you can still get there using the side streets. It doesn't mean the highway doesn't exist or doesn't have a specific function.
That's a perfect analogy. But because Lashley was so influential, his work really delayed the widespread acceptance of localization in the field of learning and memory for decades, until about the 1950s.
So it was a major roadblock.
It was. But eventually the evidence for localization just became too overwhelming to ignore. And this brings us to the modern era of validation, where we can actually see these principles in action.
And some of this validation is pretty wild. Let's talk about awake brain surgery.
This is the work of pioneers like Wilder Penfield in Montreal and later George Ogeman in Seattle. These guys were neurosurgeons, primarily treating patients with severe epilepsy.
And they had to keep the patients awake during surgery.
They did. The brain itself has no pain receptors, so you can numb the scalp and the skull, open it up, and the patient can be wide awake and talking to you.
That is simultaneously terrifying and amazing.
It's necessary. Before they would remove the small piece of brain tissue that was causing the seizures, they needed to make sure they weren't about to cut out a critical language center, so they would stimulate the surface of the cortex with a tiny electrode while the patient was talking or naming pictures.
So say apple, zap, and the patient just stops talking.
Exactly. If they zapped a spot and the patient suddenly couldn't speak or use the wrong word, they knew, okay, don't cut there, that's Broca's area. They were able to map out language function with millimeter precision. They confirmed everything Broca and Wernicke had hypothesized, but in living conscious human beings.
And they found new stuff too, right? It wasn't just confirming old maps.
Oh, they did. Ojimon, for example, discovered that the insula, a region of cortex that's folded deep inside the brain, is also essential for language. It showed that the network was even more complex and distributed than Wernicke had thought.
And then we get the technology that lets us see inside the brain without cutting anyone open. PET scans and functional MRI.
This was the absolute game changer of the last 30 years. Now we can see the brain light up based on blood flow and metabolic activity. We can see which parts are working when you're doing a specific task. And we can get really, really granular.
There's a study in the notes about thinking versus doing.
Yes, this is a classic imaging experiment. They can put you in a scanner and see that just reading a word on a screen activates your visual cortex. Hearing the same word activates your auditory cortex. But if they ask you to just think about the meaning of a word without saying it or seeing it, a specific different area in the left frontal cortex lights up.
So we can physically see the location of abstract thought.
We can see the neural correlate of generating meaning. It's incredible.
Okay, here's where it gets really interesting for me. The bilingual brain. This is something I've always wondered about. If you speak two languages, are they stored in the same place?
This is a classic case study in brain plasticity. It really shows how experience shapes the brain's physical structure.
So Hirsch and Kim did these fMRI studies comparing people who learned a second language as kids versus people who learned it as adults.
That's right. They looked at early bilinguals who learned their second language before age 7 and late bilinguals who learned it after age 11.
And the difference is physical. You can see it on the scan.
You can. It's fascinating. In the early bilinguals, their native language and their second language activate overlapping, basically identical regions in Broca's area. The two languages share the same physical real estate.
But the late learners, the people who learned French in college,
They have two separate adjacent regions. If you learn French when you are 20, your brain has to set up a new little shop in Broca's area specifically for French grammar and production, right next door to your native English region.
that explains so much. It explains why it feels so much harder to learn a language as an adult. You're literally having to build a new addition onto the house rather than just using the living room you already have.
Exactly. But here is the really interesting nuance. Wernicke's area, the comprehension center, doesn't show this separation. For both early and late bilinguals, the meaning of words is stored in the same place.
So it's strictly a motor and grammatical separation in Broca's area, not a meaning separation.
Precisely.
Speaking of grammar, we have to talk about the universal grammar experiment. This one just blew my mind.
This is the work of Musso and Moro. They wanted to test Noam Chomsky's famous idea that all humans are born with an innate instinct for grammar, a set of universal rules that underlie all human languages.
So they taught people fake languages.
They did. They taught native German speakers two types of artificial languages while they were in an fMRI scanner. One type of language followed the rules of universal grammar rules that are common to all real human languages like Italian or Japanese. The other type deliberately violated those rules. It was structurally impossible, an unnatural language.
And the brain knew the difference.
The brain absolutely knew the difference. When the subjects were processing the real artificial language that followed the universal rules, Broca's area lit up, just like it would for German or Italian. But when they tried to process the impossible language,
Nothing.
Broca's area was silent. It didn't even try.
That implies Broca's area isn't just a generic learning machine. It's a specialized filter. It's biologically hardwired to accept certain types of grammatical structures and reject others.
It strongly suggests that our capacity for language is, at its deepest level, a biological instinct constrained by the physical anatomy and pre-wiring of the human brain.
Okay, so we've covered the cold stuff, logic, language, anatomy. But what about the warm stuff? Emotion, personality, the soul, if you want to call it that.
Right. For a long time, the right hemisphere was ignored. It was called the non-dominant or minor hemisphere, the silent partner.
Because it didn't control language for most people.
Right. The left brain does the talking. But we now know the right brain provides the music of language. This is a concept called prosody.
Prosody.
It's the rhythm, the intonation, the stress, the emotional tone of voice. If I say I'm fine with the smile versus I'm fine with the glare, the words are identical, but the meaning is completely opposite. The right hemisphere is what decodes that emotional meaning.
So if you have damage to the right hemisphere in the equivalent spot to Broca's or Wernicke's area,
You might get a condition called aprosodia. You might speak like a robot in a complete monotone, or you might lose the ability to understand sarcasm or irony or humor in other people's voices. You take everything literally.
The text mentions that the right hemisphere is key for irony, metaphor, and wit.
It gives language its color, its richness, its social nuance. Without the right hemisphere, you're just reading the transcript, but you aren't hearing the performance.
And what about raw emotion itself? Fear, joy, anger.
That is also localized to a surprising degree. We know the amygdala, a little almond-shaped structure deep in the temporal lobe, is central to processing fear and strong emotion. If you electrically stimulate it in a patient, they report feeling intense, unprovoked fear or rage.
There's a bizarre example in the notes about the subthalamic nucleus.
Oh, yes. This is a target for deep brain stimulation to treat the motor symptoms of Parkinson's disease. But neurosurgeons found that if they stimulated this specific spot, it can induce sudden, uncontrollable, and infectious euphoria. The patient starts laughing uncontrollably. They even reported increased libido.
Just by zapping a tiny cluster of cells deep in the brain.
It's a stark reminder that these complex feelings are biological events. And we see this even more clearly in certain kinds of epilepsy, specifically temporal lobe epilepsy.
This is the Dostoevsky epilepsy, right? He supposedly had it.
Sometimes called that. The effects are profound. During a seizure, the ictal state patients can experience complex phenomena like déjà vu, vivid hallucinations, intense fear, or even really just ecstasy. But the interictal phenomena, what happens to their personality between seizures, is even more fascinating.
This is the personality shift.
Yes. A significant number of patients with this specific type of epilepsy develop a characteristic personality profile. They often show a loss of sexual interest, but a dramatic rise in hyper-religiosity, hypermoralism, and a strange lack of humor. They become intensely serious, philosophical, and preoccupied with cosmic questions.
That is just wild. A wiring glitch in the temporal lobe can make you hyper-religious.
It suggests that our entire personality structure, our interests, our humor, even our spiritual inclinations, are deeply tethered to the normal firing patterns of specific brain regions.
Which, of course, leads us to the ultimate puzzle. The thing that's doing all this experiencing. The I consciousness.
This is the last frontier. And some of the most profound and frankly disturbing insights we have come from studying split brain patients.
This is the work of Roger Sperry and Michael Gazzaniga.
Correct. These were patients with epilepsy so severe and intractable that it was life-threatening. As a last resort, neurosurgeons would cut the corpus callosum, the massive bridge of 200 million nerve fibers connecting the two cerebral hemispheres.
So the two halves of the brain can no longer talk to each other.
They are completely isolated, and the result was two independent conscious minds cohabiting one skull.
Give us the example, because this is the part that keeps me up at night.
Okay, so remember, the left hemisphere controls language in the right side of the body. The right hemisphere is nonverbal and controls the left side of the body.
Right.
There was a patient holding a book in his left hand, reading. Well, he was reading, meaning his left hemisphere was reading. The left hand is controlled by the right hemisphere. The right hemisphere cannot read and has no interest in the book. So the right hemisphere got bored and commanded the left hand to put the book down.
Meanwhile, the left hemisphere, the one that can read and was enjoying the story, is like, hey, I was reading that.
Exactly. The patient's left hemisphere would be audibly confused, saying, why did I do that? There were cases where a patient would try to button a shirt with their right hand, and the left hand would come up and start unbuttoning it. It was a literal conflict between two separate wills.
That is horrifying. It's like something out of Dr. Strangelove,
Right.
the alien hand syndrome.
It fundamentally challenges the very idea of a unitary self. And Gazzaniga's great insight was that the dominant hemisphere, usually the left, acts as the interpreter.
What does that mean?
If the right hemisphere does something weird that the left hemisphere doesn't understand, the left hemisphere will instantly make up a plausible-sounding story to explain it. It confabulates. It creates a post-hoc narrative to maintain the illusion that I am a single, coherent person who is in control.
So the self is just a story the left hemisphere tells itself to make sense of all the signals it receives from the body and the other brain systems.
That is one powerful and slightly unsettling way to look at it.
So what does this all mean? We've gone from fluid-filled tubes to a complex, modular, parallel processing machine. Where are we now?
We have moved from the 19th century view of the brain as a blank slate or a single lump of jelly to understanding it as a complex computational machine made of discrete, interconnected modules. It is both serial and parallel. It is both localized and distributed.
Biology and psychology have finally completely merged.
They have. You simply cannot understand the mind without understanding the organ that produces it. They are two sides of the same coin.
So here is the final provocative thought to leave you, our listener, with. If the self is a construction, if cutting a single bundle of wires can split you into two distinct people, if a small lesion can take away your ability to understand a metaphor or change your entire personality and religious beliefs, what is the I really?
That is the hard problem. That is the challenge for the future. We have a pretty good parts list now. We're getting better at the wiring diagram. But we still do not know how the objective firing of specific neurons translates into the subjective, private, first-person experience of being alive. How does an electrochemical signal become the feeling of seeing the color red, or the taste of wine, or the emotion of love?
The ghost in the machine is still a ghost.
It is. Figuring out the neural correlates of consciousness, how the machine generates the ghost, that is what the next generation of neuroscience has to solve. It's the last great frontier of biological science.
Well, on that existential cliffhanger, I think we are going to wrap up this deep dive.
It's been a pleasure exploring it.
Thanks for listening. Go take care of your brains.
Indeed. They're all you've got.